we are starting with the experiments now, which were carried out to prove whether DNA is a genetic material or not. So the first experiment or the first step which proved DNA is a genetic material or not is Griffith's experiment. So the first experiment or the first step to prove DNA is genetic material or not is Griffith's experiment. He carried out his experiment in the year 1928. He carried out his experiment in the year 1928. So before going to know what this experiment, conclusions and everything about this experiment, first we should know what Griffith has exactly used to carry out his experiment. He used two organisms. That Griffith used two organisms to carry out his experiment. What are those two organisms? He used Streptococcus pneumoniae. He used Streptococcus pneumonia, and the second organism he used is mice. This Streptococcus pneumonia is a bacteria which can cause a particular type of pneumonia in mice. Apart from this, the Streptococcus pneumonia do exist in two forms. It exists in two forms. First form is it exists in smooth strain. It exists as a smooth strain. At the same time, it exists as a rough strain. It exists in two forms. First one is smooth strain, and the second form it exists is rough strain. The name itself indicates smooth strain. That means in this bacteria, the outer surface of the bacteria is covered by a smooth membrane. This smooth membrane is called capsule. Whereas coming to the rough strain, in the rough strain, the outer surface of the bacteria is not surrounded by this capsule. Hence it appears like granule-like structures. Hence it is called rough strain. Another important feature of the smooth and rough strains are, this smooth strain is called virulent strain. Because this smooth strain is having capsule around its outer surface. This capsule gives pathogenicity. To the bacterium. That means this capsule is responsible for causing disease in organisms. That means this capsule is the main reason for causing pneumonia in the mice. So the smooth strain is surrounded by a capsule, hence it is called virulent in nature. Why it is virulent in nature? Because the capsule which is present in the smooth strain is responsible for pathogenicity. Whereas coming to the rough strain, where absence of capsule, that means the rough strain is not surrounded by a capsule around it. So this is a virulent. In nature. That means it don't have an ability to cause disease in the mice because of the absence of capsule. So here in case of smooth strain, capsule is the main reason, or the capsule is responsible for pathogenicity. The same capsule is absent over here, hence it is a virulent in nature or don't have an ability to cause the disease. So here Streptococcus pneumonia occurs in two strains. One is smooth strain, the other one is rough strain. Apart from these two strains, among these two strains there are number of varieties. Among the smooth strain, there are number of varieties like S1, S2, S3 and so on. At the same time, among the R strain, there are number of varieties like R1, R2, R3 and so on. Among all different varieties of the smooth and rough strains, Griffith used one combination. Griffith used S3 and R2 strains. Griffith used S3 and R2 strains. He has used the rest of the strains also, but the final result has come with these two strains. The final result has come with these two strains, S3 and R2. So let us see how we carried out his experiment. So case 1. In the case 1, he used smooth strain 3. In the case 1, he used smooth strain 3. As we know that the smooth strain is virulent in nature. Okay. When he injected smooth strain into mice, as we know the result, we can expect that smooth strain is virulent in nature. That means it had an ability to cause pneumonia in mice. So when smooth strain 3 injected into mice, as we know that definitely the mice will die. The mice will die. So the mice got died here, and the results as per our expectations. Case 2. Case 2. In the case 2, he used R2, rough strain 2. As we know, rough strain is virulent in nature. It don't have an ability to cause pneumonia in mice. So when R2 injected into mice, the mice survived. The mice survived. So the result in case 2 as per our expectations. Coming to case 3. In the case 3, he used smooth strain. He used smooth strain, but he used the smooth strain with heat killing. He used heat killed smooth strain in the case 3. As we know that smooth strain is virulent in nature, but here we use it is heat killed S3. That means this 3, which is virulent in nature, has been destroyed by exposing to heat at certain temperature. When it is exposed to a certain temperature, the capsule which is responsible for pathogenicity got destroyed. Now the S2 converted into a virulent. So when it is injected into mice got survived. We can expect the result that definitely the mice will survive, because here it got converted into a virulent. Okay. In the case 4, this is surprising. In the case 4, Griffith used 2 combinations which got the same result. So in the 4th case, Griffith used 2 combinations which got the same result. So here among the 3 cases, which got the same result? In the case 2 and in case 3 got the same result, that the mice is survived here. So he used R2 along with heat killed S3. Along with heat killed S3. When he used a combination of these two and injected into mice. So here the result we can expect that R2, which is a virulent in nature, don't cause any kind of disease to the mice. At the same time, heat killed S3 when injected into mice also no problem. Why? Because in the case 3, when heat killed S3 injected into the mice is survived. So definitely we expect the same result here in case 4 also. The mice should survive according to the case 2 and case 3. But the results are like this. Mice died. Mice died. This is a surprising result to the Griffith. That results wouldn't reach the expectations of Griffith, and it gave a very surprising result. Mice got died in case 4. This is a question mark to the Griffith. he didn't understand what exactly has happened to the case 4. According to the above cases, here mice should survive, but here the mice got died. He came to a conclusion that after carrying out the case 4, he came into conclusion that something got happened here which make the heat killer S3 into virulent S3. So after the case 4, he came into conclusion that either heat killed S3 may have converted into live S3. This is one possibility. And the second possibility is the R2 have converted into S3. There are two possibilities for case 4 result. For case 4 result, these are the two possibilities. Either heat killed S3 may have converted into live S3, or else R2 which has been converted into S3. So he came into conclusion that either this or this should happen. That means here transformation has happened. Transformation is nothing but changing in the form. Here heat killed S3 has been changed into live S3. That means changing in the form has taken place here. Here also R2 which is in one form has been changed into S3. That means transformation has been taking place in case 4. That is the reason why the mice got died. So that is why we are calling this experiment as Griffith's experiment of transformation. We are calling this experiment as Griffith experiment of transformation. So finally the conclusion is not so clear even to the Griffith that whether DNA is a genetic material or not.